Hello, everyone, and welcome to this week's NeuroTools Webinar. Today we are joined by Dr. Jeffrey Grethe, and he will be going over NIF and DkNET, making research tools fair. We're very excited for this week's webinar, and we're very happy that you're joining us. If you have any questions, please feel free to type them into the group chat. If they can wait till the end of the session, we will answer them at the Q&A at the end. If you'd like them to be addressed in the moment, we can also do that as well. I'll raise the question to Dr. Grethe. We do ask that you mute yourself and keep your video turned off during the session. If you need to elaborate on a question that is being addressed, feel free to unmute yourself as your question is being addressed. Without further ado, I'd like to hand the microphone over to Dr. Grethe and get started. Thank you very much.
Thank you, Devin. What I'll be talking about today are two projects, the Neuroscience Information Framework and DkNET, and the concept of making research resources fair. Most people are probably familiar with the FAIR concept in relation to data. The data should be findable, accessible, interoperable, reusable, and then also there's an additional R, in terms of recognition or attribution. However, the FAIR principles themselves, apply not just to data, but really apply to resources, knowledge, and information. So, the information about what resources, what model organisms, what antibodies were used in a research paper, need to be accessible. They need to be discoverable. You need to be able to take these and work with them together to develop, various dashboards or analytics. And all this information, needs to be able, to be reused when others are doing, their research studies. So there's, a whole, suite of, tools and potential uses, for information, knowledge, about research resources, in addition to data. And so, one of the big things that we're dealing with is just a deluge of information about, these resources and about available data and information that's out there. And it's really difficult to access, collate, and filter through this information that's in the public domain if you're a researcher. And so NIF and DkNET, were both established in recognition of the need to basically interconnect, researchers and their communities with these, large pools of resources, data, and information that are relevant to their research. And both NIF and DkNET provide a single point of entry for discovering, this type of information, and hopefully that will increase, speed and efficiency of their research processes. And so a little bit of history. In Neuroscience Information Framework was started in 2008. There were a number of developments that went on with NIF. DkNET was funded then by the NIDDK at NIH in 2012. And the infrastructure that was used to build NIF became more generalized to support different communities. So in addition to DkNET, there are a number of additional initiatives, that are all powered by this common infrastructure, which we're calling SciCrunch. And basically, all the projects that sit on top of this, have their community-specific views of the data and the information that's contained, within the infrastructure itself. And so one of the big goals of the projects, as I've just described, is really to connect researchers and resources. So basically, NIF and DkNET provide a search portal, to provide, to find community-vetted research resources, be that materials, data. There's access to hundreds of databases that we pull this information from that's accessible through these systems. We allow for more personalized search and display of results based on the community that you might be coming in to the system from. And we also provide the ability to push information out, so be an information hub for the community itself. And we also provide support for some of the resource providers, that have information within the system, and I'll talk, through a number of these different areas. And so what I wanted to do first was to give people an overview of sort of the information and the way that one can sort of see what DkNET and NIF both have to offer. And so what I'll do is I'll walk through a number of use cases, sort of switching between NIF and DkNET in terms of the examples I give, just to give you an overview. And to get started, I'll be talking about 3 different types of information. So when we look at the information that the systems contain, we have community information, which is information that's been curated by a community curator or community manager that has information relevant to that community from community-specific resources or types of information. We then have the collection of all resources, and this consists of more than 200 databases across biomedicine. And then we also have the literature in terms of providing search through the literature. So it sort of provides a one-stop shop to come in and look at very different types of information across a large number of areas. And so starting first, we'll be looking at the community resources, which is on the left, and you'll notice that the community resources are organized in a community-specific way in terms of categories that are defined. When one moves to more resources, here we divide things in terms of the data or resource type they are. And in that sense, across all these 3 indices, navigation differs a little bit across the indices. And so this is something that we're constantly doing, user testing with, trying to figure out better ways, to present all this information to users as they come into the system. And so starting in community resources, we'll look first at, searching for rodent models for type 1 diabetes. And so if one goes to DkNET, at dknet.org, one can type in type 1 diabetes, select the organism box, and basically hit search. And what that will do is then bring back, a number of records related to models related to type 1 diabetes. One of the things you'll notice in front of all the names of the resources, there's a little checkbox, and this allows you to add information to various collections. So if you're trying to get information on antibodies of a certain type, one can do a search, and one can then start adding, antibodies that you might be interested in to to a named collection that you provide in the system. And so this is, one way, that we, provide the ability for users to, manage information, within the system. And then on the left, you'll notice there are a lot of options in terms of, how you can refine the search, either by focusing on specific sources or by looking at various subcategories, in this case of model organisms. We also provide some other options, one of them being a category graph. So when you do a search, you can basically look at, what types of information are returned by that, and you can drill down through the category graph and then select, the specific entity that you want to filter down to, and it will take you and will filter that search for you. We also have the ability to look at when data was updated, to see if new records have come in from a various source or around a various category, and these types of options, allow you to do updates, and I'll mention a little bit of that later on, to see what information has updated either from a particular resource or related to a particular search or related to a resource. Next, if we move from community resources and move to all resources, in this case I'm going to be looking at NIF, searching at Alzheimer's disease. Again, we go to NIF, put in the information in the search box, and what you'll notice is that in the prior example and here, you'll see that the term gets quoted, and that's related to the ontologies that back the system itself. So, one of the areas of development is to bring together community ontologies, so information from diseases from the disease ontology, anatomical structures from Uberon, molecular information from CRBS, and bring that in to help people in terms of providing the ability to select these terms when one does a search. But what it also does, is it allows us to sort of enhance the search itself. And so in addition to the ontologies within the system itself, we also have a terminology dashboard where we can go in and explore these terms using the user interface. This is something that we're rolling out right now, so this is very, very new, but it builds off the NeuralX platform, which we had set up in the past through NIF, and this is the new version of this, within the new portal framework. It allows you to go in, look at information, look at how terms are related to one another, look at various annotations, and then we also have the option, to put in references. So where has this term been referenced, for example, within a data source, and we can maintain those within this dashboard. So again, this is a new feature that we're going to be rolling out this fall, but wanted to give a little preview of that. So again, when we do the search, you'll notice that the search term also includes a number of different synonyms related to that, and when we search all the information, we provide different ways for people then to filter that information in terms of what they would like to see. And so we have some very high-level categories that someone can select, and then we have sort of a second tier of categories which are a little more focused, and then below that we give the list of sources that have been returned with information related to the search terms. And so if you look at the full list of sources, for this query, you'll see that there are a large number of sources from the ones that we pull information from that have some information. And if I'm looking for looking for data in this case, I can select data. I can then further sub-select to look for expression information, and then have those sources returned. And then once I select the source, let's say I want to go to the top one here, ArrayExpress, I can select that, and it will take me into a table view of the information. So if you noticed from the community resources section, we sort of had a more Google style snippet view of the information. We also provide these table views of the data and of information on the resources. And so here, there are a number of different options one has. We even have ways to sort of visualize some of the information a little better. So when you have a large resource that has a large number of results, sometimes it's hard to see, okay, well, what organisms are involved here? So we also provide things like word clouds, which come down, and you're able then to select information in the word cloud. So for example, Alzheimer's disease, well, I didn't know that, that zebrafish, had, some data related to Alzheimer's disease, but when you click on that, you'll actually be returned, records, from ArrayExpress, with Daniel as the organism, and yet, there is a lead exposure protocol which does have, some linkage to Alzheimer's disease in terms of this being a model organism for that. So, and again, this is one way that we can look at the data. We also have facet graphs themselves, so you can look at how the information is again distributed across the different columns, and so this is another visual way to get in to that information. And as standard, we provide the facets on the left as well for you to drill down into that. And now if one wants to sort of transition to the literature and some of the features that we provide there, so you can select on the top, you can move from data to literature. with your search, and then we provide a search across PubMed. We also bring in search across some of the full text of the open access literature, which you can find under the options tab here. And then we also bring in, for example, altmetrics around the literature. And so altmetrics will provide information on how this particular paper is being shared. For example, readers on Mendeley, or how many times has it been mentioned either through social media, and then also provides information on how does this rate with other scientific publications. And then we also provide some simple analytics in terms of publications per year across your search. And Alzheimer's, of course, is a disease that is continually growing in terms of the number of publications that we see. Okay, so now sort of after giving that quick background, sort of the different areas and places where we have a lot of information, we're now able to begin thinking about, well, how can we actually take all this information that we have indexed and collated across these resources and really turn them into an information network that links and provides information about these research resources. And I just sort of have to apologize. My mailer has been downloading mail, and I guess people are sending me viruses, so those messages keep on popping up, but I'll keep on canceling them. So when one looks at an information network for resources, there's a whole collection of information that we're pulling information about these resources. So if it's a data set that has a specific place where one gets that, there's a DOI, there's an identifier potentially for the data set. Resources themselves, how do we identify resources? Resources have information. There are papers that are linked to resources and data sets in terms of resources being used within a paper that potentially creates or delivers a data set. And this research is supported by funding from governmental agencies, from nonprofit foundations, and there's associated grant numbers with this. And all this research for a particular study is supported by the authors and the researchers involved in that. And there are identifiers for authors as well. So this is the ORCID. So we have this whole collection of information, which with these sort of sets of identifiers, again, there's not a single identifier for all digital artifacts, because identifiers have specific purposes. So again, papers, digital object identifiers is the standard way to identify those. Authors are identified by the ORCID. But these identifiers and the relationships between them allow us to build and start to build a network around these resources, and provides the ability to basically, discover and examine sort of this landscape through different views. So if, from a funding agency perspective, in terms of the grants they support, what resources are produced, which data sets are produced, also allows, credit to be given to the producers of a resource, of a data set. We can also then track provenance, so you can see how resources and data sets were used, to create, resulting papers and, resulting conclusions coming out of those papers. And one can actually start tracking sort of how, protocols, for example, might be related to one another. And then also allows one to trace utilization and impact of these various different resources. And, to facilitate broad use of biomedical assets, platforms like NIF and DkNET, in terms of, this information that I sort of quickly previewed at the beginning, really provide a way that we can sort of mediate this, the complex world of, where all this information is and how this information is tied together. And so what I'll start describing now is how we are pulling those pieces together to really, make research resources fair, and really start building and allowing users to interact with this resource information network. And so, the first part of this is identification for the resources. So Dr. Anita Bandrowski has given a prior talk in this webinar series on the research resource identifier. So I'm going to just do a couple slides, if there's anyone new on this webinar, covering the concept of the RRID, but then I'll transition into how we're using that and linking that into the other information that we do have. And so the Resource Identification Initiative was something that was kicked off by NIF, early on, where we got journals, editors together to figure out how we can basically better identify research resources in the literature. Problem is that authors didn't provide enough information to unambiguously identify research resources, and it's also very hard to find all studies that used a resource or had information or comments about a resource that was used. And this is critical for reproducibility, for troubleshooting, and also for getting the latest information about a resource. And so, working with journals, we have been, pushing this out, and adoption of journals where RRIDs, are being utilized has grown and continues to grow quite substantially. We're now over 400 journals with RRIDs, and for the 2018. That's looking to grow to somewhere in the range of 600 to 700 journals. So this continues to grow, and is something that has been made possible by both NIF and DkNET. And so NIF as a project, as I described earlier, established this infrastructure for aggregation of information across distributed databases, and also created comprehensive registries for antibodies and research resources such as databases, software tools, research centers, and other types of resources. And so that core information then was further developed by DkNET, and I mentioned at the beginning of the talk sort of the generic and customizable version of the NIF platform, which we call SciCrunch, but that really allowed us to create these specific views on top of the aggregated data and information that we had. DkNET also expanded the aggregation of information that we have within the system, and then also has begun developing user-centered services around these identifiers. But through some additional collaborations with Hypothesis, which is a nonprofit which is developing an annotation layer on top of any web document, so any HTML page, one can annotate, and these annotations can contain lots of different types of information. So what we've done is we've developed SciBot, which reads a paper and brings back information on the RRIDs that are found in the paper. So here you'll notice that there's a research resource identifier that identifies, in this case, a model organism from JAX. And then Cybot, using information that's maintained within our systems, is actually able to place an annotation on that document, that provides, information, on that resource, and then provides links back, to the information, contained in JAX. And so we're able now to actually, online, add information, additional information to the publication using this annotation framework. And this information is now also being made available through Crossref, through event data. And so again, starting to build up sort of this information network of, in this case, how research resources are tied to the publications, and the authors, of that publication. Another area that I described was the resource registry. So this is where we contain information about software, different databases. And so this is a registry that we've been maintaining since the very early days of NIF. And we've been tracking how these resources are mentioned in the literature. So we do have the ability to text mine and crawl the open access subsets of literature that we have access to. And one of the issues there is that a lot of content available through journals is not available for text mining. It's getting a little better, but this is still one area which we can, which we hope will open up further down the road, so we can pull in, more of this information. But for example, for ImageJ in this case, we have core information about the resource. For any user who wants to see when ImageJ is mentioned in the literature in terms of mentions that we can pick up, you can subscribe to new mentions. So if you have an account, you can go to any resource that you're interested in, and you can subscribe to to updates on that resource. If you're the owner of a resource, you can also claim ownership, and as an owner, you can also look at the various types of mentions of the resource. You can edit those, you can contribute those, and you can also get this information about when we pick up mentions of the resource. Now, in terms of the resources in the registry, we also maintain relationships to other resources. So for in the case of ImageJ, ImageJ has a number of plugins. It's related to some other software resources. It's listed by a number of other catalogs and registries, and so this is additional information that we track, about resources. Again, building up, this network of information, around each resource. I did mention the references, and here, we have, various types of mentions. So we have, different levels of confidence, from verified references to, references that we've picked up in text mining, to, references, from, RRID specifically cited. And what you'll get for each reference, if there is a snippet of text, we'll provide that snippet of text showing how, ImageJ in this case was mentioned. And we also allow user feedback, so, for some of the lesser confidence mentions, we allow users and owners to basically provide information if the mention of the resource is true or not. And this is all downloadable. You can access this through the site. Now, using the different relationships and the different mentions, we're able then to start doing a number of analytics on top of this data, and to start actually providing some value-added services for the user. So in this case, switching to another resource, so the Nuclear Receptor Signaling Atlas, because of the mentions that we have, we're able to actually look at, co-mentions. So what else is mentioned, with NERSA? And here you'll see things, such as, NCBI, you'll see PubChem and IUFAR, so all things, related to NERSA. And then we're actually able, to sort of provide information on, the resources, most frequently mentioned and that might be related. And so we provide that as information, for example, for software tools or databases, related resources which might be of interest to the user. Another benefit of tracking these resource mentions is it makes it very easy. Again, the information about resources again is buried in the full text, so it's difficult to collate a lot of that information in terms of how people are describing a resource, what are their comments about a resource. And so by being able to pull out the mention text around the resource, one can actually start seeing how, for example, in this case the NIDDK Central Repository is being used by different research groups. So it provides an easy way for people to look at information and mentions about a resource from the papers. Now, once we have that all sort of in place, so we have the registries, we have the proper identifiers, we're able now to build dashboards and other analytics on top of that. And so in this case, we have a resource information dashboard, which is something that's being developed right now. It's not publicly available, but will be shortly. And here, one can see, for example, all the resource mentions, with the identifiers in the literature. And one can then start viewing this in different ways. So one can look at the literature, the paper itself. One can go then to the paper, and within that one can actually then see, okay, well, what are the all the resources that they used in this publication? What were the associated grants in terms of funding that was used in terms of these resources. And going further from the users, the paper itself, one can also then start looking at, okay, well, publishers can get information about which resources are used in their journals. So this is something that might be of value for the publishers and also for funders in terms of which resources are utilized with their funding. So again, once we start building up this information network, we're able to start layering on top these analytics and different views on top of the data that might have use for different constituents. So in terms of fair resources in support of the researcher, so what are the tools and services that we're providing for the researcher? And so one of the things that is available in terms of the basic searches that are there is the ability to save a search and then have the ability to send out information if there's new information from a search within the system. So any of the searches that I walked through earlier in the webinar could actually be saved, and then you can set them to email you alerts if there's new information. And so this is one way to sort of provide ability to sort of keep up to date with particular resources or searching for certain types of information. As I mentioned, within the resource registry itself, one can subscribe to mentions. Users can track their resources. So for example, if you use a number of software tools in your research, you can actually add them to a collection, as I mentioned earlier, and then you can subscribe to new mentions from those resources. and that can provide you updates in terms of where that software was used or any other type of information that might be published about that software tool or resource. And taking that one step further is that once you do have resources in a collection, one can actually start generating resource reports. And so these resource reports will allow you to collect information, for example, on the various cell lines, model organism, antibodies that were used in a certain research study. And this is useful in a number of different contexts. One of them is the ability to basically provide an authentication report about the various resources. So for example, we're all familiar with some of the issues with cell lines and contamination of cell lines. And so what these reports basically provide is the ability to collect information about the resources that you're using. We can then pull information from the various information resources that have information about those cell lines, and we can collate it in such a way so that we can make it very easy to say, okay, well, this cell line, has been discontinued. There could be a mention in the literature, that mentions something about that cell line, and so we can actually collate that information and put that in these reports. And again, this is also a relatively new feature that we're doing testing on and getting feedback on. But right now, what we're doing is we're allowing resource reports to be generated, and once you have your collection of resources, you can actually generate a report, and that will be stored. And in the future, we're also looking at the ability for people to add their own validation data, and this is all in support of NIH's sort of push in terms of improving rigor and reproducibility. And the infrastructure that I just sort of described and some of the tools really for the first time allows the easy dissemination about problematic resources before a study is going on, even during a study, and then after once they are in the biomedical literature. So tying sort of this type of information dissemination to what I mentioned before about hypothesis in terms of being able to add annotations on top of the literature, here what we see is that we can use that same type of framework to highlight and provide information about, in this case, a cell line, and some comments about what type, what's the potential issue with that cell line. And so again, these annotations, even though this might not be mentioned in this paper itself, by aggregating this information and then adding an annotation on top of this page, it provides up-to-date information to readers in line with the paper that they might be reading. So again, this is a way to sort of bring all this information, not in terms of having to come directly to NIF or DkNET, but layering this information where the researchers are spending their time, in this case in the literature. Now, one of the other pieces I mentioned to the puzzle of this research information network is tying this into the creators of the resources, the publishers of the papers, and this is where integration with ORCID comes in. And so again, ORCID is an organization that provides identifiers for researchers. Within our collected resources, one can associate your ORCID ID with your account. And one of the next steps that we're looking at is how can we actually push information on resources about a researcher to ORCID. So right now, ORCID is allows you to aggregate and collect information about your publications. But what we'd like to be able to do and start doing in a more automated way is then also if you are responsible for a resource, being able to add that information also to ORCID, so that we're able to basically sort of build out again the connectedness between this whole collection of information on the researchers, the resources, the data, the publications.
Hey, Jeff.
Yep.
I'm just going to jump in for a sec. You have a question in the chat.
Okay.
They're asking if you could elaborate on using, quote, reproducibility tool for antibodies, maybe like a pipeline to check out who used it and its publications.
Right. So within the dashboard that I showed earlier, you could actually search for a particular antibody by its RRID and get a list of all the publications. That will then lead to the authors that the researchers involved in those research studies. So again, we're starting to develop a lot of these different views into these networks, and that would be a way to look at potential if researchers are using the same set of resources, sort of a group where other researchers could go to find information or get information about the use of those resources. So again, I've shown some sort of initial steps that we're taking, building on the information that we have available, but there's a lot more that sort of we're planning to roll out around the analytics and the way to sort of provide information around the resources. Hopefully that sort of addresses that question. And then, one last thing that I'd like to, basically describe very quickly is